So if you're like me and you have a really difficult time understanding things in class, or just need a quick refresher on a video so you can hear it audibly, then I hope this video helps for you. It's a video about the gram staining process and what the difference between a gram-positive bacteria is and a gram-negative bacteria is. So to start, I'm just going to talk about the gram staining process, and then I'm going to talk about how we differentiate a gram-positive bacteria and a gram-negative bacteria and the difference between the two. So the first step in the gram staining process is that you have your slide with your bacteria on it, and the first step that we do is we're going to stain it completely with crystal violet. Let it sit for a certain amount of time. It depends on what your lab protocol calls for. Whatever time it calls for, let it sit on that. And the process of this, or the purpose of this, I should say, is so that the stain is going to stain all of the bacteria cells purple or blue, depending on your crystal violet, because they can be a little bit different colors. Step 2, we're going to add iodine to the same slide that already has the crystal violet on. And again, we are going to do this and cover our slide completely in it. And the cells / bacteria are still going to remain that blue or purple color. We're not changing the color with the iodine. And our third step, what we're going to do is we're going to add alcohol. We're going to completely wash away the crystal violet and iodine. So you just want to slightly elevate, or tilt, I should say, your slide, and use like a water bottle with alcohol solution in it. And it's going to slide off the crystal violet. Don't be super rough on this because you could actually wash away your bacteria. And the purpose of this is that your gram-positive cells are going to stay purple or blue. And your gram-negative cells are going to be colorless at this point because we just washed out all the color of them. And our last fourth step is to do a counter stain. So we're going to use saffron to counter stain, because the saffron is going to absorb into the gram-negative cells, and the gram-positive cells are going to stay this purple bluish color. And once we put the saffron on, the gram-negative cells are going to become this pink color. So when we are trying to differentiate, the difference between the two, the big thing that we're going to see is that gram-positive bacteria are going to be this bluish purple coccus-shaped bacteria. So after we've done the gram staining process, if we see that, then we're going to know we have a gram-positive bacteria. If you see something that's a pinkish rod color, then you'll know that we have a gram-negative bacteria. So I just want to show you a picture that's really helped me understand this and why the counter stain and the crystal violet, how they kind of get absorbed in it. So I just want to show you this really awesome slide that I found. So here we are. We have the gram-negative. What this is looking at is how the stain actually goes in and it gets absorbed. So on a gram-negative, you have a very thin layer. This purple right here is the peptoglycogen. And because it's so thin, we're not gonna, that purple washes right out. And when we counter stain it with the saffron, it's going to become pink. And on the other hand, our gram-positive that we have here has such a thick peptoglycogen that it is all that purple is going to just absorb right into it and love it. It's going to be super difficult to wash out 1, 2, 3, 4 layers versus 1 layer on our gram-negative. So that's why the difference is, because gram-negative, again, has a very thin peptoglycogen layer, and gram-positive has a very, very thick gram-positive peptoglycogen layer. So I really hope that this video helps you to determine the process of gram staining and what the actual difference is between a gram-positive and a gram-negative bacteria. Thanks.